He used to live in Toronto. He came here first to work at the Ontario Cancer Institute, and he used to be our principal investigator at the legendary Amgen Institute. He was our very own Arnold, except ours cured people instead of terminated them. Joe Penninger's work not only touches on osteoporosis, on heart disease, on autoimmune disease, but apparently, without exaggeration, it may even yield the fountain of youth and the cure for dying. But we let him go, and now he visits us from time to time. So Joe, thank you for coming back. Come up and tell us what you've been up to in Vienna since we saw you on our stage last. Joe Penninger.
So it's good to be back home. I lived here for 13 years and I really liked it here. And I will tell you a little story what we are doing in Vienna and why we are doing it, and maybe something which might have relevance. I find it, this talk now was really interesting. So I'm a genetic engineer who makes genetically engineered mice. And I will show you why this has these little dots, because I will probably show you a movie about flies' eggs, something we got into recently. So, but this is my thing of desire. So the Austrian Academy of Sciences came to me and said I should come back. They give us 100 million Canadian dollars to build a new building. So, and the thing which was really interesting is I could build a place with infrastructure where we just bring in good people and just let them work. We just bring in bright young kids from all over the world. We hired now more than 100 people already from 40 countries who joined us in the last 1.5 years in Vienna, and just give them all the tools to fool around a little bit and do some research. And basically give them the intellectual, and I should also say spiritual freedom to just worry about the ideas and just create something new. So that's a typical fly, a genetically modified one, as you can see. But we actually set out and said it's basically impossible to study genes, so we know genes from many organisms, but how can we actually figure out the function of the genes? So flies have around 15,000 genes, so we set out to make 30,000 transgenic lines in the last 2 years. Invested lots of money, invested 40 people who work day and night, and the library is finished yet. So it's basically a library of the whole genetic universe of a fly. Flies get cancer, so we can basically study within 1 year all genes which control cancer. Flies have energy, metabolism, so we can actually just do screens if they can fly or not fly, to find genes which control energy. Flies can get addicted to cocaine. So within 3-4 months, we basically can pull out every single gene in the genome which controls cocaine addiction. Flies have pain, so if you squash a fly the next time, so please think about it. And Barry Dixon, a young geneticist from Australia, joined us. And he came to me and said, I want to study and find all the genes which control sex. So I thought, that sounds really good. Let's do it. So this is actually a male having sex with a female here. I don't want to show you the movie, so it might be censored. And this is a fly brain, and actually this fly brain is labeled with every neuron you see now is neurons which control sex. So this is a gene Barry found called fruitless, and this fruitless gene controls if a fly has sex or not. And these are actually the brain cells which are doing it. And just to show you how this is looking, so I'll show you a little movie, if it works. I've never done this before. Oops. So here you go. So you see, this is actually a female, and this is a male, and the male is chasing after it. So we call it also the skirt chasing gene. And what you can actually see, the flies this guy is touching it, it's dancing for the female, and it starts singing. So sometimes you see the male puts out the wing here, and the wing pull out is actually a love song to the female. So basically every fly which sits on the kitchen table, so if you kill them, you might interrupt a courtship procedure. So now the amazing thing was, if he changes the fruitless gene in females, so it turns out only males make a protein from the fruitless gene, but not females in the brain. So only males have the fruitless protein in the brain. So he genetically engineered a female which makes the male protein in the brain. And actually what you see is here courtship behavior from one female to the other. With a single gene change, the females start to behave like complete males. They start singing and dancing to other females, chasing them. So this is a female which sings a love song to another female. So this was quite exciting because we knew there are single master genes, for instance, which control organ development. So it turns out in flies, and we don't know if this is in humans or in mice, it might be more complicated, but there are single master genes which control complete behaviors like courtship and sex behavior, and singing and dancing to other organisms. This, for instance, is and you can take it a little further. So these are mating chains. So actually, these are males chasing other males, because if you take off the fruitless gene, basically make a fruitless defective male, what happens is you have males which want to have sex with other males, and they start chasing each other, and they have these chains, and they basically chase one male after the other one. And the same happens if you turn on the fruitless gene in females. Females start to chase other females. So a single gene in the brain only, without affecting the body or the hormones of the fly, completely rewires the brain so that males start to only chase males and females start gain complete male behavior. Many of you might have lived through this. SARS hit town, SARS hit Toronto, and all of a sudden we ended up working on SARS. And I will tell you a little bit about the story which is developing. So some years ago, we did again a fly screen, and this is actually a young fly, a larvae, and this will become cells which become a heart. So flies have beautiful hearts. They also go into heart failure when they get older, and we had these markers called Eve or Tin Man, and we looked for mutants which, for instance, didn't look normal here. And when we looked for these mutants, we hit on a gene and which cloned it, and we called it, and others cloned it, ACE2, angiotensin converting enzyme. And some of you might know this because essentially everybody who has hypertension gets blockers for the brother of ACE2, which is called ACE. And so we were curious if flies have a second ACE2 gene, what is actually ACE2 doing in mammals and in humans. And so we made a mutant mouse again. And turns out what ACE2 is doing, it controls heart function, because this is a normal heartbeat, so this is basically echocardiography, so what you're looking at is a heartbeat of a mouse. And when ACE2 is knocked out, these guys go into progressive heart failure. So in essence, we have now a model for angina pectoris and progressive heart failure. So when we published this, somebody pointed out that ACE2 was not only in the heart here, but it was in two other organs. And one was the kidney, and we still don't know what it's doing in the kidneys. And another organ was the lung. And so we started to look in the lungs, and all of a sudden got completely stunned by a paper coming out from a group in Harvard, which identified my favorite protein, ACE2, as the potential molecule SARS binds to infect people. So this is the SARS virus. It had these beautiful little spikes coming out, and these little spikes basically would bind to this protein we like, ACE2, we had found in our fly screens, and would basically control that the virus can enter our cells and would infect us. So it was not really clear if ACE2 is actually the real receptor, because there are other papers which came out, so it was very unclear, and I will show you now what this thing is doing. So that's the SARS story. I find it very interesting because also for social reasons, so at the end, around 800 people were infected. 10% of them died. It was, you know the story here, there was a movie recently in Canada. And how SARS kills, it's basically, it lets people drown in their own lung fluid. So these people get lung edema, they cannot breathe anymore and basically suffocate to death. So this is a major problem, and we thought, let's put one and one together. Maybe ACE2, which is the thing SARS uses to infect us, has some role in lungs because we knew it's in the lungs. A student of mine built an intensive care unit for mice to study acute lung failure. And to make the story short, turns out if you shut down ACE2 in a mouse, the mice get absolutely unbelievable acute lung failure. So ACE2, this protein SARS is using to infect us, protects our lungs from blood leaking into the lungs. So the next question then was, can we use this to explain SARS? So the major issue, of course, is why the hell became SARS such a lethal virus? The other coronaviruses out there, some brothers of SARS. If you get this, you have a little cough, you're happy after 2 days. So why is it that SARS killed 10% of the people? And mind you, under circumstances of intensive care of modern medicine, so it still was killing many people. And the first thing is we actually hooked up with a group in China, the group which was last in the headlines because 2 people in the lab died. They got SARS infections because the virus spread through the labs. And to basically the first message I want to tell you is ACE2 is indeed the essential SARS receptor. You shut down ACE2, the SARS virus can do nothing anymore. So that's good news. So we now finally know the molecule SARS uses for entry into our body. The second thing, and this is data and I won't bore you anymore, turns out the SARS infection completely shuts down ACE2 in the body of a mouse. Basically what SARS does, it makes us look like an ACE2 mutant animal, a little mouse. So the question then is, maybe it was lucky that SARS actually happened and we can learn from it. Because if SARS uses the protein which protects us from lung failure, then we could just turn around the whole thing on the SARS virus, make the protein as biotechnologically developed protein, put it in a mouse, and we should actually have the capability to rationally treat SARS-regulated lung failure and death. And so we did this, and that's exactly what happened. So this is SARS-induced lung failure. We put down the pathway, we rescue it, so we have a rational therapy for SARS now. And of course, the next question is, can we actually use this for potentially treating avian flu, or Spanish flu, sepsis? So we put this protein, and sure enough, it works. So the short of it, we believe SARS showed us a completely new medicine to treat the disease which was previously deadly and kill people. So sometimes killer viruses are not bad. And I will stop with this here. This is this beautiful building in Vienna. Who has ever seen it? It's called the Sezession. There's a very beautiful Klimt painting inside, the Beethoven frieze. And it's hard to read now. But on the top it says, every time has its art, and all art must be completely free. And so I think every time has its science, and all science must be completely free. So we live in a great time, so let's use our science to do something good for people. So thanks.